with that being said, we can go ahead and start our officer Q&A. So let's go ahead and start with Rahul.
Hi, Dr. Srinivasan. Can you tell us a little bit about your academic trajectory, having received graduate degrees in peptide chemistry and nucleoside chemistry? And my second question was, can you share about the choices you made and what ultimately inspired you to become a medical researcher?
Yeah. If you go back to look at peptides and nucleosides, peptides are what I call small proteins. And let me, there's a note here with F. Peptides are small proteins, and nucleosides are the basis for all DNA and RNA. So the DNA in our body comes from RNA, and so nucleosides are all derived from, the DNA and RNA are all derived from nucleosides. So if you get expertise into these two, then if you learn how to manipulate them. My idea was, and also I had two very good professors in my PhD program and in the postdoctoral program. They are probably the instrumental in, I don't think they taught me like in a class, just the way they made you think. They didn't teach you, hey, do this or do, no. They let you figure it out yourself. So they created a kind of a circumstance or an atmosphere around me so that you grow yourself. So you want to be independently thinking. And the best lesson everyone taught me is how to connect the dots. That's, I always tell everyone, if you want to be in this research or any research, connect the dots. The reason is, for example, when I want to design a compound, I have to know what are the properties of the compound. That comes from people in the tumor biology, where they look at the properties of the tumor. So we know what kind of compound that you can use to target it. And then when you want to use nucleosides, sometimes we can design nucleoside to, one of the compounds we did at Stanford is how to detect the rate of growth of the tumor. Because if it's a slow growing tumor, you have a better chance of curing it. If it's a fast growing tumor, you have to use a certain other product. So with these two in mind, these are the two areas of chemistry you have to get expertise in if you want to be in the medical field. At least that's my opinion. I don't think that's one. And also, you can also learn how to inhibit certain enzymes that increase the tumor growth rate. But actually, I didn't get into nuclear medicine until much later, after postdoctoral work. My first job was how to inhibit certain enzymes that replicates the tumor DNA, how to arrest them. So that was my first job. So I hope that these are the reasons why I said this has got to be the interesting field to get into. So is that a satisfactory answer?
Yes, thank you. Another question I had was, were there any particular experiences or moments like throughout your training and work experiences that stood out to you as key learning moments?
Key learning moments. Oh, there were times that we had so many results, we had no clue how to interpret them. It's like, we know we had certain compounds, and these things have like 5 or 6 possible structures that That's what it was, but it's something like mathematics, you can say, but we ended up with like one equation with three unknowns. So it was for nearly a month or two, it was just a giant puzzle. Even a couple of professors that I was reporting to, and these are the biggest learning curves, that like a curveball that thrown at us to learn. And also other learning moment was the first time in the neuroendocrine tumor field, the first compound we went into clinical studies. One thing in this field is we do a lot of animal studies before the FDA will let us go to the humans. And still there are surprises that human body behaves slightly different from all the best animal models. And the first compound that we had no clue as to why the compound behaved differently. And so it just pretty much, we had to go back to the drawing board. But we learned what the structural feature in the compound that made that problem. So we learned it quite a bit, and we were able to figure it out. So these are all the things that you're going to face, but you have to do it. Hello?
Thank you.
Yeah. These are the biggest learning things that you have to do. This is going to happen sooner or later. Not the same way, but different things are going to happen like this.
Hello, Dr. Srinivasan.
Hi.
So I just wanted to ask about like, how was your experience as a consulting professor at Stanford? Like, more precisely, what did you enjoy teaching in the medical field, and like, what are some of the challenges you faced?
I got a couple of things in my notes. Guys, one, I had a lot of fun, and also it's pretty demanding. When I start, I know many of them because when I was in Bayer, I was working in Germany. We had some collaborative projects at Stanford. So when I retired and moved back to California, Sam Gambier at the time asked me whether I would like to become a consulting professor with all my industry background to help students in postdoc, which is, perfectly fine, no problem. From my home here in El Dorado Hills, I used to go to Stanford once a month for a week. And I enjoyed going there, talking to students. I'll give an occasional seminar, not teaching a class, I say. But also, one of the things I was able to do is, since I've been in the field for longer than anybody in the department, since the early 90s, nearly 25, 30 years, and that was an emerging field at that time. This radio labeling peptides, we are the first ones to do it. And so one thing I had in my brain all the time is all the history and all the failures. What doesn't work, because nobody publishes failures. So I pretty much have in my brain storage what works and what doesn't work. So I can easily tell people, point out, I think you should look for a different method, different procedure. Or what are the properties of the compound that we should incorporate in the compound in order to be successful. So these are the things I was able to give them options, and I still do. I work with, so far the department was engaged only in detection of diseases, but now we are into therapy, and the only one who have done it. So I work closely with 2 or 3 people to come up with some new therapeutic agents. It may take 3, 4, 5 years, we don't know, but at least we got started, and the university was nice enough to give enough funding to get going. So that was the plus. My goal is I can also learn at the same time.
So
But
like, as you mentioned, the fact that you like were had many like trials, and then so also the fact that there's also many trials in order to first test on humans too. So how exactly, what motivates you to like to go continue even after like failed trials and like failed experiments?
I always tell people is, if you're afraid of failure, this is not the business you should be in. It's going to happen. If you want positive results every time, I think you should always tell people you should open a grocery store. At the end of the day, you have some money. If in this research business, if you succeed, say 20-25% of the time, you are doing extremely well. This is part of the business. And once you know, fail, and then you try to figure it out and you succeed, that's addictive. You get so high when you succeed. There is nothing like that. It's altogether a different feeling. So you have, if you're afraid of failure, please, this is not the business to be in. You have to find something else to do. That's my stern advice to anyone. And that's the same thing like Akash mentioned towards the end of doing astrophotography. It took me 2 years to learn how to do everything, but failure is part of the game. That too, unlike in this one, sometimes you sit outside looking at all the sky, do everything until midnight. There are days you get nothing. If you're going to give it up, yeah, you can, but it's the same story. So my brain, I think, got used to, if we fail, just, you fall down from the horse, you dust yourself up and get back on.
So it's like the feeling of success that motivates you?
Yeah. When you spend 30-40 years and you have one compound, one success like that last year, according to statistics, because the compound is marketed by Novartis, during our first 2-3 years before Novartis took over, we treated about 2,000 patients in 4 centers. And nearly 3/4 of them are living for 6-7 years. I don't think you can beat that. That kind of feeling that, hey, something that I made with my own 2 hands are in these people, and they are living, and few of them wrote to me. Oh, there's nothing like that.
Thank you.
That's what keeps it going. What do you think at the age of 75, I'm still doing it? That's the reason.
Thank you.
Yeah, so kind of as a segue from like what Tejas was saying, do you think you can tell us a little bit about your kind of research successes and failures, and like what helped you choose where to focus given all the options available, and ultimately choose an area where you felt you could have a major impact on patient treatments?
How do you, the process of, okay, let's say, for example, you mentioned that my work was in the neck chin. So why did I go to choose that? Why not something else? One of the things is first thing to do is whether I have all the background information necessary to treat that tumor. Background information means properties of the tumor, because the compound we are designing is like a key that fits into the lock all the tumors on the surface of the tumors, they have certain proteins that are present only in those tumors. Maybe it's present in some other organs in a smaller amount. So we are designing these compounds that target those tumors. Okay? And therefore, so you need all this information that's published in the literature, because there are researchers who work in tumor biology. They don't know any chemistry. Their goal is to look at all the tumor biopsy samples and figure out, what are the properties of the tumor, so that when they put it out there, somebody like me and others come along, use that information to target those tumors. This way, when we do these things, we don't do this in isolation. I don't work in isolation. I need the help of those people, and also the help of, I know about the biology and isotope radiation, but I'm not expert in doing that. So my colleagues are people who knows about the isotopes, how to handle them safely. And then there are scientists who test these test these compounds in animals. So you need the cooperation of everybody in order to succeed. So at no time I want to say, well, it's all one man's effort that got you where you are. There are so many, and then there are people who can manufacture them, and there are physicians who tell, okay, what's the dose I should inject? That's altogether a separate field. We can make guesses based on animal studies, but these are the people who decide how to do that. And so this is how the whole thing works. But one thing you have to do in every one of them, can you tell me what the last part of the question is?
Yeah. So ultimately, like, how did you decide what area you felt you would have the most impact on?
So we figured when we started this project, we had all this information about neuroendocrine tumors. So we said, okay, this is the first test case for our technology, that when we combine peptides to target those tumors with the radiation, specifically targets. Once that works, then we can extend this to other tumors. So that's how we chose that target. And also we had the collaboration of the tumor biologist who discovered all those ones. And then he gave us the ones about the prostate tumor, and that another group in Johns Hopkins did that work. Because I left Mallinckrodt, Mallinckrodt did not want to continue in this business after that. And so the next product came out based on the same technology that we invented. And also we invented the first group in the world to use lutetium metal isotope, 177 radiation isotope. We are the first one ever to use that isotope. Until then, that isotope was just a real curiosity item. Now everybody is using lutetium 177. So we figured that out because it is good for 7 days. Its half-life is 7 days. So we can manufacture that within the country in one location, the whole product, and ship it overnight. And it's still good for 6 more days. So we figured all these things out because we had the 4 people that in my group that worked together. The only other thing I had to tell you is, when I went to Mallinckrodt, I was in a management position. One day I walked into my boss's office and said, is that okay if I leave the management position and go back to the lab? So they gave me a scientific position so that I can go back to the lab and do it myself. I had no problem getting rid of my management title, go back to the lab with 3 others, so that I get the joy of doing this myself. Plus, I got nearly 20 years of experience with peptides and nucleosides. I can do it faster than anybody. So to me, that was the. So it's not just I was able to design and make the compound. Those things are done with my own two hands. Not when I was 20 or 25 or 30. I was 55 when I did that, went to the lab to do it. And I loved every minute of it.
Wow, that that's pretty crazy, actually.
Yeah.
Yeah,
I can remember I was talking to Jeff Dunn. He said, "Are you sure you want to do this?" I said, "Yeah." Because there's no other. That was the biggest fun I ever had.
Do you think like did it affect your ability for like getting future jobs, do you think, or do you think it was just?
I'll tell you, when I wanted to, when Malincrot did not want to pursue this again with the next target, I had a call from, I know quite a few people in the industry. You know, these kinds of words spread. The head of research in New Hampshire at the time, the Bayer had an office in New Hampshire. He called and said, "Srini, if you want this job, it's yours." I got, you talked about my resume, I worked in 5 different companies. At no time I applied for a job. Every time they knew my publications and what I've done. Somebody called me and said, "If you want to come, it's open." I didn't have to even apply for a job. And then when I went to Bayer at New Hampshire, they wanted to consolidate all the research in Germany. And so my boss was in Germany, called me. He came to New Hampshire and said, "Hey, why don't you move to Germany? You have 10 times more facilities and the group to do it." I said, "Fine." I packed up and moved to Germany.
Jeez. How long were you in Germany for?
I was there for 4 years. That was my original contract. And then he was telling me, "Why don't you stay for?" Things were going good. He said, "Why don't you train a couple of more people before you leave? And you can stay for 4 more years." No problem. I said, at that time, I'll be retiring, so I just retired. And Stanford asked me whether I can become part of their faculty. No problem. And I don't get paid in Stanford, but why am I doing it? Because it's a lot of fun. And after retirement, I still made 6 publications in major journals.
Wow. That's impressive.
The last 6 publications came after retirement, being a consulting professor at Stanford.
Jeez. Okay.
So that was, I do it just, plus it keeps my brain active. It's not like tomorrow I have a big meeting about discussing about, we're going maybe go for growth, but we want to design a peptide that targets certain tumors entirely in a different way. We're going to try, our chance of success, I'll say 50-50. It might take a year. But one thing, my advice to anyone who gets into not this healthcare field, no, there are so many healthcare fields. Always have a backup plan. Don't throw all your eggs in the same basket. Always, if this compound doesn't work, what's the next? What's the next? You always have that options in your pocket. If something doesn't work, you should know to take up the next. Because like I was telling you earlier in the conversation, that the first compound we went to clinical trials didn't work out. No, we could see the tumor, but there are other problems. It didn't clear the body quickly. And we were able to figure out why. So we had some backup compounds. The other reason is you have to put the best compound into trials, because once the trial starts, you're talking millions of dollars spent in clinical studies, because you're doing it in 10 different places. You're talking we have to pay the doctors, we have to get the instruments, everything. The clinical trial can easily cost of isotopes, add everything. My salaries, everything. And once the trial starts, we're spending millions of dollars. We can't substitute compounds at the last minute, because then you have to do all the toxicology studies, you have to write the FDA applications, you have to get FDA approval to go to clinical studies. So you cannot, at the last minute, substitute another compound. So it's a very long process. So you have to go with the best. For the same neuroendocrine tumor, we had another compound that's about 10-15% better, but we are so far along in the clinical studies, we just let it go. We cannot do anything, because you cannot spend another $20-30 million to start all over again. That's life. But once in any health, I don't mind if any of you get into any healthcare research, as long as it helps the patient, just one extra step. One extra step. You're doing a good service. That's all you can ask for.
Yeah.
Because when I was one day sitting in my office at Stanford, two of them walked up to me and said they're going to quit their PhD program because they know how to do all these imaging studies and they're pretty good in computers. One of the things in this business I'm in, there are 2 or 3 different companies that make all these instruments. And when you want to become a physician and go through all this, nuclear medicine physicians, during their training, they get used to one instrument or the other because hospitals have different brands. And sometimes there are quirks between different brands. So these guys wrote a computer program so that they started a small company. They wrote a program that no matter which brand of the instrument that you use, say the imaging instrument, they all look the same. So they wrote a software that does that, and they're making more money than many professors. So they came up with their own idea, took the risk, and made this work. So it's all up to individuals. So as long as you can make the patient life just a little better, that's all you should ask for.
Yeah. Okay. I think with that we can go ahead and get into Akarsh's question now, which is kind of touching base on what you talked about a little bit.
Okay.
Yeah, hi, Dr. Srinivasan. My name is Akarsh, and my question is, we're all really interested in your work treating tumors. What did you learn along the way, and what was the most challenging part of this type of work?
What's a challenging part of the work?
Yeah.
Challenging part of the work was when, before we started in this business, the only isotope, the radiation isotope that was always used was iodine-131 isotope that was used to treat several tumors. the way they did that was they just inject that into the body and then it'll go to the thyroid to treat thyroid tumors. So when we started looking for any other isotope, why we selected lutetium-177, because we can put this compound in a cage and then attach it to the peptide. How to keep the peptide properties intact while attaching another metal to it? That was the most challenging part of the chemistry. So that's the chemistry. At that time, nobody knows how to do it, how to design a compound to attach this isotope to the peptide. That was the most challenging part, at least in my opinion. And then a few minutes ago, I was telling you about the first compound we did. Didn't work out. Somehow or other, compared to the animals, in the humans, we figured out that the compound folded into a different three-dimensional structure that we didn't expect at all. So we had to get blood samples from the patient, because hospital always collect blood samples of the patient after the compound was injected. So we had to get blood samples from Holland, because that's where the studies were done, and then figure out what's happening and then fix it. That we were able to do it in 2 months. Those 2 months, I don't know whether 3 or 4 of us ever went home. That was but once we figured that out, it was heaven. So that's the best answer I can give you. Was there another question that you had?
No, that's it for me. Thank you. That was very interesting.
Yeah, that was there's okay. And then you have to know what it is there are some tumors you cannot treat that with this method at all. I know that because there are tumors that are radiation sensitive, there are tumors that are not radiation sensitive. And I can still remember somebody once sent me some samples from India, the doctor's reports. That person had a neuroendocrine tumor, but lacked in the biopsy sample, I could see he lacked one protein that's needed to target the tumor. So it's like, he must be so unlucky. He was a pretty famous person. He's so unlucky. Probably 1 in 100,000 cases are like that. We couldn't help him at all. And these things are going to happen in nature. And it's difficult. But you have to look at no drug is going to be 100% effective in anyone. That's life. Humans are not made exactly like, it's not like iPhone, one is exactly like the other. We are not made that way. But always put the best effort that you can. The ability to connect the dots is something everybody has to know. The information you want is there. Like I was telling somebody the other day, another division of the company that I was, they developed an imaging agent for Alzheimer's. And now we know there are reports coming out in the literature that Alzheimer's could be due to a virus infection that happened 10 years earlier. So now it gives you a hope that how to test people who are getting older for virus infection. Then you can prevent Alzheimer's. Right.
Okay, yes.
Hi.
Thank you, Doctor. My name is Tumin, and I have a question regarding how COVID-19 has had an impact on medical research, and how has it impacted how you work, and what are some of the setbacks due to COVID?
Oh, I'll tell you the good news and the bad news. The bad news is in Stanford labs, most of the labs had to be shut down for almost a year. A year is losing a year is a big deal because students and the postdocs, and then not only, I think we lost the time in the lab, and also like we keep a lot of animals, testing animals. You cannot keep them for indefinitely for a long time. So many of the animals just, they are not cheap. They are certain transgenic animals that lack certain proteins where we can induce tumors, things like that. Many of them are so expensive, they all have to be, destroyed. And the labs were not functioning, and few of the people in the lab, they showed positive tests for COVID, even though they didn't get the disease. So we lost more than a year. What's the good news? We were able to do a lot more literature work, look at the literature and try to figure out what are all the new projects we could do, have all these extra discussions within the group. So that's, otherwise we have to kind of, scrounge for time all the time. People have to do a lot of extra work. And so we were able to do all that, paperwork, writing for new grants, people were able to do things like that during that time. So that was the benefit, but really more than a year was lost.
Okay, so hello, my name is Aryan, and
Hi.
my question is, what is your perception about the advancement of medicine in current society, and what direction do you think we are headed in that regard?
I'll tell you which way we are heading, at least my opinion. I don't know whether you are following whether we have the Human Genome Projects. Then the next level of drugs, non-radioactive ones at least, will come out of based on Human Genome Project. I'm pretty certain about that. And then in terms of the technology we are using, we were able, in the last 5-6 years, people have come up with the properties of other tumors. Like, for example, triple negative breast cancer, which are most difficult to treat. And then we have, one of the things the industries don't do very well is, if there is not enough big market, they won't do it. That's always the problem with the industry. So one of the things we looked at is, we made a list of tumors because they come to the university hospitals. So we are able to get all these biopsy samples of these tumors, so that I think the next 5 years, we want to use our technology to treat tumors that are otherwise no drugs are available. To me, that's one of the best things that we are able to do. That's from the Human Genome Project, and then I wrote another for just a second. Time loss. Oh, the Human Genome. There was another one. Yeah, and on the brain research, and also there's a big European effort is going on to map everything in the brain. So this is one of the things they are thinking. Maybe in the future, the dementias and Alzheimer's disease drugs will come out of that mapping the brain. So this is where, and then like about few weeks ago, that because of the COVID, they were looking at the samples of the brain that they were able to find that the previous versions of the COVID virus, SARS virus, probably infected the brain, and whatever small amounts that's left, that's why it takes a long time for the disease to manifest itself. So now people are going to look at, can we use a sign of a blood test or some other mechanism to see, like, after a certain age, you get a prostate test and things like that. So whether we can prevent these old age diseases by screening for virus samples and things like that. So if that happens, that will be very nice. But it may take years, I don't know. I'm not an expert in that kind of area, so at least that's what I read. The thing, I think, if you want to get into this healthcare field, I think there are some newsletters, magazines, I think you should look at. And also the other one, I always urge people, there is a streaming service called Curiosity Stream. If you get a chance, there are excellent documentaries about healthcare. And also it tells you about how the human body works. You can pick the field you want in healthcare. There are documentaries there. It's an excellent learning tool.
Okay, thank you. I think with that, we can go ahead and start a member Q&A. So for people that may have questions in the audience, you guys can go ahead and either raise your hand or message them to me or any of the other officers, and we can go ahead and ask it for you. So we'll give it a minute or two. Yeah, Rahul.
So Dr. Srinivasan, I had a question. Aside from medicine and everything you've done in like the research field, can you tell us how you manage your stress and any anecdotes or from experiences you've had in the past?
One anecdote I've given. After finishing high school, I wanted to enter the college. I had no idea what to study. In those days, there are, like you have all this ideas, like you're talking to me, and in 60s, there's nothing. So what my father did was, he went to a Catholic school, Catholic Jesuit college. So he took me to the same priest that taught him. And he said, that's how they taught the priest. He told my father to go sit outside. He said, I want to talk to your son. They always taught, son, you study chemistry. That's where the future is. I said, okay, I'll study chemistry. I had no idea what I'm getting into. And then stress, I don't know. You know what? People talk about this. They said they do this exercise, that exercise. For I don't. I have, like, for example, after I go to, when I was working full time, I was always interested learning about space, evolutions. Space means astronomy and everything else. I've followed what NASA did since my age, from the first Gemini program in India. And I spent time looking at that. That's why the first thing after I retired is go get my own telescope and look at outer space, take pictures. Then now to spend time. I make movies of birds with themes in it. I have no problem in nothing gets to me. I have when I just leave the chemistry alone for a day or two, and I can go concentrate on something else to get my energy back. And then I have some major discussions tomorrow with 2 or 3 others at Stanford. Yeah, I spent like part of the day today to get ready for this. I didn't think about that part at all. Tomorrow morning, I'm just good as new to tackle that one. I don't think I felt particularly stressed about chemistry. Anxiety, yes, but there are days, one of the things that in this business it's very important is you have to be sure about what you're doing. Not only that, if somebody else repeats the work that you did, which has to happen, because you are doing clinical studies in 5 places, everybody should get the same results and everybody should inject the same compound. It's made in the labs in different places. It has to work right every time. So everything that you do is documented correctly. That's absolutely important. Those are the stress we put on ourselves to do it right. But that's not going to get us down. That's a necessary stress, a necessary evil that you have to do. But that's okay.
Okay. I think we have about time for one more question. So if someone else has a question, go ahead and, like I said, raise your hand, or you guys can message one of the officers privately.
I actually have a question.
Sure.
Okay, so okay. Hi, my name's Sadhana, and I was just wondering, so how do you feel about your work in the way it used to be today, in the way it's used today? Like, how do you feel about it?
Today, I think when we started it 20, 30 years ago in this field, it was difficult because there is no background information or anything else. Because we were the first one to start. We have no clue how to handle these isotopes. People have to figure it out. Now there is so much background information, but also there are more facilities in terms of there are expertise in handling isotopes, there are expertise in handling dosimetry. We had to figure all those out in those days. Now, there are, like for example, if you go to Stanford, in those days we have to buy even an isotope. We have to go to a special reactor to order them. We have to anticipate what we need next month. But now there are many facilities that produces the isotopes, many facilities to produces the amino acids. So there are commercial companies that make so many amino acids. There are machines to make peptides. So you have more and more facilities to do the job a little bit easier. So I'm expecting people will produce a lot more better results. Because, like for example, if I want to make 100 peptides tomorrow to test, I can probably do everything in 5 days. Before it'll take me 100 days. Now I can do it in less than 1/3 of the time. So these are all the, just like you go from a typewriter to a computer. That means you're also, like for example, I can sit at home and read all my journals that's in the Stanford library. When I started it, I have to stop everything that I do, have to make a special trip to the library, go through the books manually, indexing everything else. Now I can search everything at any time I want and read any journal I want. So you have all the information that you want in your fingertips, which is not the case so many years ago. Until like 20, 25 years ago, going to the library is a major job. Most of the time we got to do it on a Saturday, so that during the regular days, we can do the work. So it's a very different atmosphere to do research now than before. Sometimes, I do get upset that with all the facilities, why productivity is not as high. Because if you treat this like a job, I don't think it's going to work. People, you have to like what you do to succeed. If somebody thinks, it's a job, I can go do some 8 to 5 and it'll work out. No. You may get a paycheck, but you're not going to succeed. So it's a different mindset to succeed. So
thank you for that answer. I hope that answered your question, Sadhana.
Yeah, thank you so much.
Yeah, so with that being said, I just want to thank everyone for joining today. Most importantly, of course, thank you, Dr. Srinivasan, for making the time in your schedule to kind of give us some insights about your career and your choices. And our membership definitely has to make decisions about our own future. And your advice is hopefully going to guide some of us to continue to make our steps forward on that path. I also wanted to express my appreciation for Mr. Chow, who's our advisor and has helped us launch this organization. And I would just like to remind you guys to join us in February, where we focus our MedGives initiative actually and our work with Friends of Children with Special Needs. And as aspiring medical professionals, we have a responsibility to strengthen the social sector as well. And see you all next time.